Hello, students. Good evening, and I am sure that you all are doing good. And today I am discussing about a very important and interesting chapters, the main aspects of this chapter, that is principles of inheritance and variation for class 12 students, CBSE board. So the deeper points are in this chapter, that is the inheritance of one gene, inheritance of two genes, and then your incomplete dominance, and then your codominance. So inheritance of one gene in a monohybrid cross, two contrasting traits from a character, say of height, tall and short. So, according to Mendel's first law, law of dominance, that is, the one trait will be dominating over the other. That is, in this case, all the plants will be tall because tall is the dominating trait. And in F2 generation, after self pollination in this F1 progeny, in F2 generation, what we get? We get three tall plants and one short. This is law of segregation because the trait which was hidden in F1, which was recessive in F1, has segregated itself in F2. Fine, this is law of segregation. But what is happening here? Only one gene is being inherited in a monohybrid cross. There is no blending of tallness and shortness. There is no mix up. The tall trait is being inherited. Maybe in both F1 and F2, maybe in F2 3:1 is the ratio, three tall plants and one short plant. Fine, but the gene which has been inherited, carrying the information of tallness and also of shortness, is one only. Fine, so this is the inheritance of one gene. Clear? Now we are saying law of dominance. First law. So how this dominance takes place? In a heterozygous combination only, there are two alleles. One allele gets modified, gets changed, and now starts dominating. Suppose there is a gene which is carrying the information of the formation of an enzyme. Now, if one allele has been modified or changed, then what can happen? Normally, the enzyme can be produced, that is all right. But in the second case, the enzyme can be produced but with no function. And thirdly, there can be no enzyme. So, this is the changes which are leading to dominance. Okay, so this is how the one trait is dominating because of some modification, some changes in the other allele. Okay, so this is how dominance is taking place, and the factors. These are the factors which are called the gene, and one trait, one allele will dominate over the other in a heterozygous combination. Sometimes what happens, there is an incomplete dominance. For example, if we take colors of flowers, say red and white, and after crossing, what is happening? The red color is dominant usually, and in this case also. So in F1 generation, all the plants will have red color flowers, and in F2 generation, as the law says, law of segregation, the other recessive color, that is the white color, will get separated in the ratio of 3:1. So 3 will be phenotype 3:1. That is, 3 will be the red colored flower and 1 will be the white colored flower. So this is the phenotype. Genotype will be double R, that is a true red, capital R, double capital R, and then there will be two hybrid, that is capital R, small r, but will look red only. Two hybrid will look red only, and the fourth one will be one pure white, that is a pair of small r. So like this, symbolically we can express this. But sometimes what will happen is that the red color, which is dominant usually, is not completely dominant over white. So in the F1 generation, there will be a change. All the colors will look pink. Another color, that is pink. So this allele of pure red is not completely dominating over white. So there is another color which has appeared, that is pink. But in the F2 generation, the ratio will be the same as the Mendelian ratio, that is the one pure red, that is capital R, pair of capital R, and two pink, capital R small r, capital R small r, and the fourth one will be pure white, that is a pair of small r. But the flowers will look pink in F1 generation, all pink. And in F2 generation also, in the case of hybrid, it will be pink. So there is a change here because the trait of redness or red color is not completely dominant over the trait of white color. So this is incomplete dominance. Okay, and now sometimes there will be codominance also. Before that, I will discuss about the inheritance of two genes. In a dihybrid cross, in monohybrid cross, there were single pair of contrasting characters. Here, there are two pairs of contrasting characters. One character is of shape and another character is of color. So this is applicable to seeds. So seed can be of round in shape. Seeds can also be wrinkled. And if we take the another trait in this combination, that is the character of color, so it can be yellow or it can be green. So the combination of round yellow is dominant over wrinkled green. So round yellow seeds, again after germination, we get new plants and then new seeds, and then we see that in F1 generation the dominating combination, that is round yellow, is appearing in from all the plants. The seeds are all round yellow, and in the F2 generation we are finding that all these traits of shape and color have combined with each other irrespective of the pair they belong to, in the ratio of 9:3:3:1. So in a Punnett square, we can show these combinations after taking the gametes in the square. In at the top of the square, at the side of the square, we take the gametes and then we start combining with one another. Then we get the combinations in this ratio only, 9:3:3:1. I am sure that you all are able to draw a Punnett square and are able to express this properly and efficiently. So here what is happening, what is the purpose of explaining this dihybrid cross is that here again the inheritance is of only two genes. That is, one is of the character shape and another is the character of color. There is no blending again here. There is no mix-up. So all are separately being inherited, these two genes. So this is the inheritance of two genes in a dihybrid cross. Now we talk about the codominance, which is happening where? In the ABO system of blood grouping, particularly in the RBC cell membrane, there are polymers of sugars which protrude out, and they are having three alleles. In a heterozygous combination in human, these alleles, there are two alleles only, and the alleles of this gene, which is carrying the information of the sugars, kind of sugars are IA, the gene is I, and the alleles are IA, IB, and small I. There are three alleles, and IA and IB are completely dominating over small I. So the combination, can be two alleles only. So in a pair, so IA can combine with small I. Small I is recessive, the blood group, the sugar will be A, blood group will be A. If IB is combining with small I, then the group will be B, because I is recessive. And if IA and IB are combining, so the sugar will be both sugar A and sugar B. So that is the blood group AB, and this is the case of co-dominance. IA and IB are combining, so the blood group will be AB. Okay, there can be two alleles only, and if it is small I, then the blood group will be O. Like this, there can be 6 combinations, which the case of co-dominance is also there in blood group AB, right? So this is an example of the ABO system, which is being controlled, and the sugars, polymers of sugars in the RBC cell membrane is being controlled by gene I, which has got 3 alleles, and different combinations can occur in this combination, and co-dominance is also there in this setup. So this is the co-dominance. Students, I think the basic concepts I have talked about, and now detail you will get in your book, NCERT book. So please study them thoroughly in detail, and if there is any query, please let me know. Thank you very much. God bless you all.